Hi, in this video, I will show you how to identify virus sequences from next-generation sequencing or NGS by using a website called Galaxy. We can use Galaxy to analyze information from various NGS platforms such as Illumina or Oxford Nanopore technology. But in this tutorial, we will focus on analyzing data from the Illumina technology. This picture shows the steps we are going to use for the analysis. Imagine we have a tissue sample from a sick pig. For some reason, we suspect a virus infection, and we are planning to use NGS to identify the virus in that tissue sample. We have extracted DNA and RNA from the tissue and then perform Illumina NGS. After we receive the raw data or raw reads from NGS, the first step is to perform quality control or QC on the reads. Usually, we remove poor quality reads during this process, but we can also apply other criteria to filter the reads, such as their length or complexity. After that, we remove the reads that come from the host. Since our sample is from a pig, a large portion of the raw reads will be pig genetics. Removing these host-related reads can make virus identification easier in the subsequent steps. To identify the virus-related reads, we will use a method called virus mapping, which can be done in two main ways. We can either map the virus directly from our sample, or we can do assembly first to get longer contigs, which provide more information compared to the short reads. Now let's go to the Galaxy website. Type in usegalaxy.org. There are three main parts on the Galaxy page. On the left panel, you'll find a range of tools that we can use to analyze the data. In the middle panel, you will see the details of the tools or the data. And on the right panel, you will find the history of the data and the analysis. Now, let's name our data history first. Click on the pencil symbol here, then change the name as we want. In this video, let's name the history virus identification. Then click Save. You will see that the name has changed to virus identification. Let's upload the data into the history so that we can do the analysis. The first data we want to upload is the raw reads from NGS. To upload the data, go to the left panel, then select Upload Data. Then a new window will appear in the middle of the screen. We can upload data in two ways. The first one is to drop the file in this area. Or you can click this button to select the file. After we have selected the file, it will look like this. The selected file will appear in the window. Then click Start to upload the data. It may take some time depending on the size of the file. After it is done, the file will appear in the History section on the right panel. After uploading the data, the NGS file will appear in the History section, and if we click on it, we will see some details of the data from this window. You can click on the eye icon here to see some information about the input, the process, or the output of the dataset. Those information will be shown in the middle panel. From the format of the data, we can see that this is a FastQ file. We can edit some of the file's properties, such as the name of the file, by clicking the pencil icon here. Or if we want to delete this dataset, click on the trash can icon here. Once we receive the raw data from NGS or raw reads, the next step is quality control or QC. In this process, reads that do not meet our criteria are removed or modified. This ensures that only high-quality data is used for further analysis. In this tutorial, we will use a tool called FastP for the QC process. To access the tool, first go to the left panel, then enter the tool's name in the search box. Similar tools will appear at the bottom of the search box. Now you can select FastP from the list. Once the tool or program is selected, the settings window will appear in the middle panel. Here you can adjust many parameters of the tools so that they work properly for your job. Galaxy tools normally come with default values that are suitable for many situations. So normally you may only need to modify some parameters as necessary. For FastP, first you need to specify whether your dataset is single N or pair N. For this tutorial, the dataset is single N, so we will select single N. Then select the dataset itself. Normally, all the files available in the history will appear in the list, but right now, there's only one file in our history, which is R1, so there is only one dataset to choose from. Next, go to the adapter trimming section. By default, FastP can automatically detect well-known adapters such as adapters used in the Illumina NGS, and these adapters will be automatically removed from the reads, so normally you can use the default setting in this part. Let's move on to the next step, global trimming. Since we are working with Illumina NGS data in this tutorial, the bases at the end of the reads usually have lower quality scores, so we will need to remove the last base from all of the reads. And here's how to do it. Go to the Trim Tails section and enter the number of bases you want to remove from the end. For this tutorial, we'll just remove one base, so go ahead and type 1 in the box. You can leave the rest of the settings as they are. The default setting works just fine. Now we are ready to run FastP. Scroll down to the bottom and click the Run To button. You can also find the Run To button at the top of the panel, so choose whichever you prefer. Once you have clicked Run To, you have to wait for the tool to complete the analysis. Sometimes it could take many hours, depending on the dataset, the tool, and the traffic on the Galaxy server. Once the process is complete, the result or the output will appear in the History section on the right panel. Normally, FastP will give two files or datasets. The first one is a summary report in HTML format. This report provides a lot of useful information, such as the quality scores before and after the QC process, and the number of reads that were removed during the QC process, etc. By looking at this report, you can see how much data quality has improved. If you are not satisfied with the results, you can change the settings and run the QC process again using the original dataset. The other output from FastP is the resulting FastQ dataset, which means the reads that pass the QC process. This FastQ dataset is what we will use in the next step. But before we move on, let's take a look at the report. Click on the eye icon here. The information will appear in the middle panel. The report has many parts, so feel free to explore it. One thing I want to highlight in this tutorial is the graphs showing the quality scores before and after the QC process. This first graph shows the quality scores before the QC. The y-axis represents the quality score and the x-axis represents the position in the reads. So this graph basically shows the average quality score at each position along the reads. Normally, NGS data from Illumina results in short reads of about 150 bases, so the x-axis in this graph goes up to around 150. In general, we want a quality score of at least 30. If the average quality drops below 30, we need to remove or trim that part out. For Illumina reads, the overall quality scores are usually high, but they tend to drop as the reads get longer. The quality often drops below 30 at the last base position, so we will trim the last position from every read in the dataset. From the graph, you can see that the quality drops below 30 at the last position. Now let's see what happened after the QC process when we cut the last base position from all the reads. Scroll down to the next graph. This graph shows the quality score after the QC. You can see that at the last position, the quality scores are no longer below 30. So now, every position has an average quality score above 30, which is exactly what we want. Next, we will use the FastQ dataset that passed the QC process for the next step. If you click on the eye icon of the dataset, the details will show up in the middle panel. From the format, you can see that this is the FastQ file. After the QC process, we will remove the host sequences from our data. For this tutorial, we will use a program called HiSat2 to do that. Go to the search box and type in HiSat2. Then select HiSat2 from the list. Simply put, HiSat2 will map or align our dataset to a reference genome. This means we will end up with two types of reads in our dataset, those that match the reference genome and those that don't. HiSat2 also has a function to separate these two types of reads, allowing us to choose how we want to work with them in the further process. In our case, we will use a pig genome as the reference genome, since our sample is from pigs. We will also tell HiSat2 to keep only the reads that do not match the pig genome. This way, we can focus on the virus sequence identification in that dataset. Go to the source for the reference genome section and choose Use the built-in genome. We choose this option because Galaxy already provides the pig genome data for this too. Alternatively, you can upload other genome data to the history and then use it as a reference genome for HiSat2. Next, we will choose the pig reference genome from the list. Click on the list and type pig. There are two pig reference genomes available, and we will use both of them, but we cannot select both simultaneously. To work around this, we will run HiSat2 twice, each time using different pig reference genome. First, we will remove the reads that match the first reference genome from our dataset, and then from the resulting dataset, we will remove the reads that match the second pig reference genome. So let's select this pig reference genome first. Next, we need to specify to HiSat2 that our data is a single-end NGS data. Then we will choose our dataset for HiSat2 here. Remember, this dataset is the one that has already passed the QC process. Click on the Advanced options. Scroll down to the Output options. Here, you can tell HiSat2 to save the reads that either match or do not match the reference genome into separate files. In our case, we are interested in the reads that do not match the reference genome, so select Yes for write unaligned reads to separate file. If you also want the reads that match the reference genome in a separate file, choose Yes here. We can use the default settings for the other part, so now let's run HiSat2. With these settings, HiSat2 will generate two outputs. The first one is a FASTQ file containing only the reads that do not match the pig genome. The second file includes mapping details for each read, but we won't focus on that for now, so I will delete it to keep the history section clean. Feel free to explore that data if you're interested. I want to point out here that there might still be some pig genome sequences in our dataset. HiSat2 might not eliminate all the host sequences, but the majority of them have been filtered out. Normally, after this process, up to about 60% of the dataset might be removed. So now, we can work with a much smaller and cleaner dataset. Suppose we have removed both of the pig reference genomes from our dataset. Now we will call this dataset as clean reads. In the next step, we will focus on identifying virus sequences within these clean reads. This process is commonly referred to as virus mapping. Virus mapping can be done in two ways. The first method is to map the clean reads directly to a virus sequence database. Alternatively, we can assemble the short clean reads into the longer contigs first, and then use these longer sequences in the virus mapping process. One major advantage of adding the contig assembly in the process is the increased reliability of the virus mapping results, since the mapping is based on the longer sequences. Now let's rename the dataset to match the details shown in the diagram. There are many tools available for virus mapping. One commonly used tool is Kraken tool. First, select the type of reads of our dataset, which are single N. Then select the input file from the drop-down list. The next parameter that we usually adjust is the confidence value. You can change the value here, or you can use the slider bar here. Increasing the confidence value can make the mapping results more reliable with a slightly reduced sensitivity. You can set the confidence value to about 0.5 for improved reliability, or you can leave it at the default value, which is 0. Next, select the database for the mapping process. Galaxy offers many built-in databases for Kraken 2. For virus mapping, we will select pre-built RefSeq indexes viral as the reference sequences. These databases only have virus sequences, so the mapping results would look more simple for the virus mapping process. After the analysis is done, we will get one output file, which is a table of the mapping results. Next, we will transform this Kraken result table into a graph because it is more easier to understand. We will use a tool called Convert Kraken for this step. The Kraken table will be converted into the Galaxy taxonomy format. Then we will convert the Galaxy taxonomy into a Corona pie chart. We will need to do this in 2 steps because we cannot directly convert Kraken to a Corona pie chart. To convert Kraken table to Galaxy taxonomy, we only need to choose the input file, which is the Kraken result table. Normally, there's no need to adjust any other parameters. The default values work well with Kraken data. After the run, we will get one more file. You will notice that the table is much easier to understand compared to the Kraken result table. The first column is the name or the ID of our reads. The second column is the taxonomy ID of the reference sequences that match each read. The next columns are the taxonomy data for the matching reference sequences. In the next step, we will turn this taxonomy data into a Corona pie chart. In the settings, first select the type of file that will be transformed into a Corona pie chart. In our case, it is a taxonomy file. Then choose the file. Next, we will decide how many levels of the tree we want to display. Normally, we select Show Entire Tree. You can use the default settings for the remaining options. Now we are ready to generate the Corona pie chart. Now that we have the Corona pie chart, let's click on the eye icon to show the pie chart. The Corona pie chart shows the proportion of the taxonomy from the reference viruses that match with our reads. For example, this chart tells us that 43% of the reads that match with the virus database are matched with influenza A virus. It represents 43% of the reads that match with the virus database, not 43% of all the reads. So Kraken and Corona pie chart can tell us about the virus population in our sample. This also helps us identify which viruses to focus on for further analysis. For example, in this case, we might create a database specifically for influenza A virus, since it represents the major population in this sample, or because we are interested in the influenza A virus. By doing this, we can obtain more detailed information about the influenza A virus in our sample. The limitation of using Kraken, at least in this way, is that they only indicate the presence of influenza A viruses. They don't specify which strains of the influenza A are present. To determine the specific strains, we need to create a custom database and use other tools to perform the mapping ourselves. In the next step, we will create a database specific to just influenza A. Then we will assemble the contigs from the clean reads, and then we will map these assembled contigs to the influenza A virus database. In this tutorial, we will use data from NCBI Virus to create our custom influenza A virus database. You can follow along on the screen to go to NCBI Virus website. When you reach this page, click on Influenza A virus here. If you need information on influenza A, B, C, and D, you can tick all these boxes. For this tutorial, we only need influenza A. This section shows the number of data that match our selection. For example, with the criteria of influenza A virus, we have about 1 million nucleotide sequences, about 1.4 million protein sequences, and 7 reference genomes. On the left panel, you will see some options to filter the results. You can adjust these settings to make sure the sequences we get match exactly what we need. Keep in mind that the database will be extremely large if we include all the influenza A virus sequences available on NCBI. One thing we prefer to choose is the completeness of the nucleotide sequences. Here, you will see there are two options: complete nucleotide or just partial nucleotide. If we choose complete nucleotide, the record count is only about 10,000 records. Normally, it's recommended to start with complete nucleotide because they are more reliable than partial nucleotide sequences, and the database won't be too large to handle. But for some viruses or some situations, the records of the complete nucleotide might be too few. In that case, adding the partial sequences to the database can be a good idea. You can always start with a small database and then expand it once you get some initial results. This way, it's easier to manage and you can refine your focus as needed. So in this tutorial, we will create a database using the complete nucleotide sequences. To download the sequence, click on the download button. The download window will pop up with 3 options to choose from: sequence data, accession list, or the result table or metadata of the sequences. This time, we want the nucleotide data. Then click the Next button. In step 2, you can choose to download all the data that we just selected or download just a random sample of that data. For this tutorial, we will download all the records. In step 3, we will choose the format of the FASTA header. Normally, we stick with the default format, which starts with the accession number followed by the GenBank title. Then click Download. Next, you need to select the path or the folder to save the file, just like when you download anything from a website. Then upload the influenza sequences into our galaxy's history. The virus mapping method we are using this time is called BLAST, similar to the NCBI BLAST that some of you might already be familiar with. However, we're using our own custom database because Galaxy cannot directly access the NCBI database for BLAST functions. So right now, our first task is to create a BLAST database in our history. To make BLAST work, we cannot just use the FASTA file as is. It needs a specific file type called a BLAST database. To create a BLAST database from the FASTA file, go to the search box and type in make BLAST. Then select NCBI BLAST plus make BLAST DB from the list. This tool is used to create a BLAST database from a FASTA file. First, you choose the type of database you want to create, either a protein or nucleotide database. In this tutorial, we are creating a nucleotide database. Then, in the Select Input section, select the input FASTA file that you want to convert into the BLAST database. You can use the program's default values for the other parameters. Now we are ready to run the program. When the program is finished, you will have a file with the format BLAST-DBN. The letter N in DBN is for nucleotide database. Now that we have the database ready for BLAST, the next step is sequence assembly. There are several tools available for assembly. In this tutorial, we will use a tool called MEGAHIT. Go to the search box and type in MEGAHIT and select MEGAHIT for metagenomics assembly. After that, select the type of reads that you want to use for the assembly. Our reads are single N, so select single N. Then you have to select the file for the assembly. You will notice that the file that shows up here by default might not be the one we need. So make sure every time to choose the correct file. In this tutorial, the file that we want for the assembly is called cleanr1, which is a FASTQ file that passed the QC process and has had the whole sequences removed. The default values can be used for the other parameters. After the assembly is complete, you will have a new file in the history. Let's click on the eye icon to see the details. Each sequence will be represented by two lines. The first line provides information such as the ID of the contig shown at the beginning of the line, and at the end of this line, you will find the length of the contig. You can see that the sequences are now longer than before. The original reads are only 150. Next, we will use this assembly to map against the influenza database using the BLAST program. Go to the search box and type in BLAST. There are many BLAST programs available depending on the type of database and sequences you are working with. What we are going to do is to search a nucleotide database with nucleotide sequences. The program for that task is called NCBI BLAST plus BLAST N. So select that from the list. In two parameters, first select the file to BLAST. Next, in the Select Database section, choose BLAST database from your history. Go to the Type of BLAST section. There are 4 types of BLAST available, but we normally use the first two, MEGABLAST and BLAST-N. In simple terms, the main difference between MEGABLAST and BLAST-N is that MEGABLAST only shows results for sequences closely related to those in the database, while BLAST-N allows more mismatches between the sequence and the database. So normally BLAST-N is used. In the Output Format section, you can choose the format of the report and which parameters to include. For a simpler report, it's recommended to use the following setting. Select Tabular, select which columns. This allows you to choose the columns you want to include in the report. Deselect all the standard columns and select QSeq ID, the ID of the query, SSeq ID, the ID of the sequence in the database, pident, percent of identical matches, and length, which means the alignment length. Then go to the Extended Columns section and select QLength, the length of the query sequence, and SLength, the length of the sequence in the database. Then go to Other Identifier Columns section. Select S-Title, the name of the sequence in the database. This is the last one you should add into your BLAST report. Next, go to Advanced Options and select Show Advanced Options. Then scroll down to Maximum Hits to Consider and Maximum Number of HSP. Set both of these parameters to 1. This ensures that each query matches only one sequences in the database, which will simplify the report significantly. After the BLAST is completed, the result will be added to the history. Now let's take a look at the result table. With our settings, the first column is the ID of the assembly. The second column is the ID of the sequence in the database. The third column is the percent identity between the assembly and the reference sequence. The next column is the length of the alignment. It indicates the length of the alignment between the sequence assembly and the database. For example, in the first row, 28 means that the percent identity of 92% shown in the third column is based on an alignment spanning only 28 nucleotides, not the entire length of the assembly. Column 5 and 6 are the length of the assembly and the length of the sequence in the database. As for this assembly, the length is 321 nucleotides. The influenza sequence that matched with this assembly is 2,280 nucleotides long, and the name of that influenza virus is shown in column 7. Using a custom-built database and BLAST program allows us to obtain more detailed information about the reads and the assembly compared to Kraken. We can start with Kraken to get a rough idea of the virus composition in the sample. Then for more detailed insight, we can use assembly followed by BLAST. Alternatively, sometimes we just skip Kraken and go directly to BLAST. But in that case, a database of reference viruses should be used, which can be selected from NCBI Virus. And that wraps up our tutorial. I hope you found it helpful in learning how we can identify viruses from NGS data using Galaxy. Remember to keep exploring and experimenting with different tools and settings. Science is all about curiosity and discovery, and with tools like Galaxy, there's so much we can learn. Thank you for joining the tutorial. Goodbye.